Max.
Oh, okay. Now it's 3 o'clock. We will start our webinar. I want to say welcome to all the people. And the topic is the RETImap with the SLO image and the ERG, the precision ERG. And this is from the company Roland Consult, and we are located in the city of Brandenburg an der Havel, and you can see also a little bit from the Havel. Our focus on ophthalmology in electrophysiology is already 29 years already. And in the invitation, we write that our flagship product is the RETImap, and we produced this already, or we started with the production since 2012. And what is important, the RETImap brings the SLO image and electrophysiology together. And today we have some topics. After this small introduction, Mr. Fu Tang will explain something overview. Then we will listen to the lecture of Ahua Chiang. Then for the lecture of Chifu Wang, Song, maybe I talk not correct the name, and Ying Xu. And during the lectures, you can write your questions in the chat. And then we can, after each lecture, we can talk about the question. And in the end, we can also summary questions. I will now say to Mr. Fu Tang, please make a small introduction. I will stop here.
Okay. Yeah, hello everyone. Yeah, let me introduce our RETImap precision electrophysiology. Yeah, this is the device. The left is the RETImap mainboard. That's the SLO. SLO that can do the multifocal ERG, pattern VEP, pattern ERG as a stimulator, and they can real-time monitoring the fundus and get a good fixation for the animal. Yeah, the right is the gas field for the full field ERG and flash VEP. And other the system for the animal. The three is the QT standing is for a flat large animal, and the 4 and the 5 is for the large animal and the small animal table, and the small animal table can combine with a thermometer. And this is the function of the SLO. Yeah, it can be do the IR fundus image, and then can real-time monitoring the fundus target area. Then we can stimulate with the multifocal hexagons and or the focal flash or the checkboard for the pattern ERG and the pattern VEP. So we can do the multifocal ERG, focal ERG, PERG, PVEP for the animal. Yeah, this is a very special systems for the animal, the focal fundus function disease research. And we can, our software can set according to your personal requirements. then we can do other many special multifocal ERG and the special PERG and PVEP. And this systems can combine with the gas field to do the full field and ERG and flash VEP. And same as just know the special multifocal ERG, the full field ERG also can set for the special parameters, including all special ERG such as the ISCON, PHNR, or on-off ERG, STR, and others special red, special color ERG, and the serial fulfilled ERG. And we also apply the different electrodes for the different animals. And this is the paper result from the RETImap SLO for the multifocal ERG for the different animal. The left is from the Johns Hopkins, that's the monkey multifocal ERG. We can see in the this the central area. This is the abnormal lesion. We can saw the abnormal curve, the lower curve, and the surrounding area is the normal area as a control. Yeah, it's very good result. And the middle, the result is from the China, from Zhongshan Ophthalmic Ophthalmic Center. Today, the Professor Zhong will introduce experience for the monkey multifocal ERG. And the right is from NEI. Today, Professor Qian also will introduce their experience for the pig, the multifocal ERG. And this is for the mouse and rat multifocal ERG. From the left, we can see the above is the normal multifocal ERG, and the lower is the abnormal. This is the same mouse. Yeah, very. So we can do for the same position for the function evaluation for the animals. And when we use the gas field, we can finish our special fulfilled ERG. And this is the result from the different papers, including the different series, the ERG and the ISCON, and this is the on-off ERG from the different papers. And this is the result is for the glaucoma research. We can do so many different index for the ganglion cells function. Yeah, that's including the STR and the PHNR for the uses the gas field stimulators, and the other pattern ERG and the multifocal. Even the multifocal PHNR or the multifocal PERG, we can use the SLO system for the research. And today, the Professor Xu will introduce their experience use SLO pattern ERG and pattern VEP. So if we do the animal, the pattern VEP and the pattern ERG, the SLO is a good device. And the one good news is we have just published on a new book. Use our all Roland cases, including some of the animals experience. And you can buy this book from the Springer web. So this RETImap system is an excellent research tools for the ophthalmology. Okay, thank you.
Thank you, Mr. Flantang. This was a wide field of the users, what we can do with the RETImap. And now we have different lectures, and we will start with Mr. Haoua from the NIH with the experience. I'm very interested what we will hear from him. Thank you for coming to this.
Thank you. Thank you, Ms. Finger and Ms. Tang for the invitation. So let me start to share my windows. And I think this is one. Can you see?
Yes.
Okay. One thing now I'd like to add is that now we had a RETImap now very early on, and we have one system that is fully loaded, that including now both the multifocal ERG as I mentioned, but also including now OCT and the fluorescent angiogram and GFP capacities. So we are starting a new project that combine OCT and multifocal ERG. And so I just want to say there's more options available for RETImap if people are interested on that study. And so today I will just briefly talk about, let me just start the slideshow. What is over here? This one. Okay, so briefly, and I'll talk about what we use multifocal ERG RETImap as a way to evaluate the functionality of the cell therapy in a pig model of AMD. And as we know that now the RPE cell health is one of the major contribution to the AMD disease, both for the dry AMD and wet AMD. Those RPE cells are commonly not dead, and then it leads to eventually loss of photoreceptors and blindness. So, and this project is a bigger group that uses the stem cell to induce, become induced RPE cells, and then introduce into the eye, and then to replace this damaged RPE cell and to restore the eye function. So our goal is to now use the autologous iPSC and then to replace the damaged RPE cells. And these cells are placed on the scaffold, biodegradable scaffold on membrane, so that with the time, then those scaffold can be dissolved and then leave with intact RPE cells. We choose the pig as a model because the pig eye has similar size as a human and has been commonly used as a model for the human eyes, both for surgically and for the physiology. And the size of the implant is about 4 by 2 millimeters, which is pretty large, cannot be accommodated by the common rodent eyes. But whereas the pig eyes provide a really suitable size for both practical reasons and as a follow-up. And I also want to mention that the study has been summarized in this paper, and the data as a part of a package submitted for FDA for phase 1 trial, which are starting right now at NEI. And here is an image of pig retina OCTs, and they have laminar structures. I just want to mention that pig eyes is dominated by a high contribution from cones. So their cone ERG or photopic ERGs are relatively large. So that's also an advantage for a multifocal ERG recording of photopic conditions. And here is a kind of picture of setup we used for this RETImap to record multifocal ERG in a pig. And so if we replace just one small pieces of the RPE cell, then one can think maybe if you use a focal ERG, maybe the way to see how that functions on that part. But commonly when you are shining a light into the eye, a large portion of the response, because the only recording of the total response from the cornea, and a large portion of the response is not from that stimulated region, but from the scattering of light entering into the eye. So there are a lot of contaminations from unstimulated region. So especially when the stimulus is small, it's very difficult to tease out the response is from the region that you wanted, or it's just from the background stimulations. So that's why the multifocal ERG is used. And it utilizes this kind of pseudo-random M sequence and stimulating different part of the retina in a pre-recorded order or M sequence. Then using a mathematical method, then you can derive the response from each stimulated grid. And the Roland this MATLAB instrument is nice, is that it combines the fundus image with the same optical path for the stimulus. So you know exactly where these lights are stimulated on each grid, and they are corresponding to the fundus locations. And from these mathematical calculations, you can derive the waveforms for multifocal ERG response. And this waveforms resembles the flash ERG. But since they are not different, the flash ERG is true recordings, whereas this multifocal response is mathematical calculations. They use different terminologies. So this first negative response is called N1 for multifocal ERG, whereas flash ERG is called A wave, then followed by positive B wave. But in here it's called as the P1 positive peak, first positive peak. So for the flash ERG, for the light adapted photopic flash ERG, this A wave has large contribution from off bipolar cells, because in the cone pathway, you have both on and off pathways in mammalian retina. And off pathway is have a very big contributions to the flash ERG. And similarly, current thinking is that this N1 is also has a large contribution from off bipolar cells, but has also contributions from the cone photoreceptor itself, but it's relatively in a small proportion. Whereas this positive peaks is from mainly the on bipolar cell response. So here is you kind of example of using this RETImap multifocal ERG recording from a pig, normal pig retina. And pigs are, again, has a cone-dominated region called the strix near this octave nerve head and between those vascular structures. And in here show a 2D summary of the amplitude for the P1 positive response. And here is 3D map. And you can see it's recapitulate this visual streak regions very nicely in the pig eye. And we can push to the limits using a much dense grid for this the pig eyes. And again, you can see it has a high density or spatial resolution for the response. And also we can still pick up the response difference between visual streak and the other part of the retina. So the model what we used for the AMD in a pig is induced by the laser, but with a caveat. So the laser is now just shining the light into the eye, and then the eye absorbs the light and produce the heat. If you slowly cooking or just continuously use the laser, then the cooking the retina slowly, the damage would be much larger in the depths. So you can induce a lot of damage to different type of cells. from RPE to photoreceptors and to the inner retina. But if you limit this pulse, light pulse, to a very short brief, then you can create a local heat that only damage a specific part of cells. In our case, we want just damage the RPE cells. So we use this micropulse laser to then just give it at a different time and do this to create very brief and specified lesions in a pig eye. So here is an example. On this left side is a fundus image. And you can see after now laser damage, this is the RPE cells are damaged and leaving a more bright view on the fundus. And here is the OCT image of this scanning here, showed this damaged area versus neighboring healthy area. In this case, now the surgeon actually missed one part in this damaged area, both showed on the fundus image and on the OCT image. Then we did multifocal ERG recordings on this eye, and the results are shown in here. And you can see this, now first of all, this is the neighboring undamaged area has high amplitude, whereas this lasered area response is much lower. But this small region that missed with the laser damage is actually the response is preserved. So this indicating that the multifocal ERG recorded by RETImap really had a high spatial resolution to reveal the small difference in retina functions. And this is a protocol that we used to induce for the RPE formation in the dish. And then afterwards, we placed on the scaffold and then introduced into the pig eye. And here show a brief video of the surgeon doing the surgery. And here is damaged, the laser damaged area. First, you coming in and cut a small hole and then create a bubble, a retina separations from retina to the RPE cells. And afterwards, we do a rectotomy to create an entry point. And then we put a special tools to cut a little bit wide opening, so we can able to allow the large piece of RPE cells as shown in here in the black, and then this curved one allows the curvature and not blocking view directly there, and then through this hole to introduce this piece of RPE cell on the scaffold then to the subretinal space. So afterwards, then the retina is flat, and then keep it attached back to the eye, and then we can do the follow-up at different times, and we also do the OCT and multifocal ERG recordings to look at, and here is the surgical OCT to look at the retina, where the retina and how is distance of the scaffold with the retina. And here's the example. And this before this normal baseline of the normal eye, and this is right after the laser damage to create a model. And afterwards, in part of this laser area, we have a scaffold is implanted, and then this is damage control, and then neighboring is health area for normal control. And here are two more examples and show a fundus image of this transplanted RPE sheets. And here is the OCT image. And here's another view at a different time. So here is a histological evaluation of transplanted cells and their survival. So this is the normal area, the OCT area. This is sections of this normal area stand for PNA for the cone out segments. And RPE65 standing for the RPE cells in the red showed in here. But on this razored area, you can see the RPE cells are largely damaged, which induce also loss of photoreceptors in this region. But in this area that transplanted by the human RPE cells, which has a marker for STEM21 as showed, L121 is showed in here, and with the RPE65, which is again marker for the RPE cells, you can see these are the human RPE cells. They are survived the surgery, and also the inside the pig eye. And also, they also support the survival of the cones on top of these transplanted RPE cells. And here is again the histological evaluation of function of these transplanted RPE cells using this rhodopsin as a marker. So in normal RPE cells has phagocytosis capacities to engulf the tip of the rod photoreceptor, which then shows this RPE, the rhodopsins in the in the normal RPE cells. And a similar function can be also be observed in transplanted these human RPE cells in the pig eye. So here shows multifocal evaluations of these transplanted eyes. So this is the pre-lasers, it's normal the multifocal ERG function on the eye. And here is the after lasers, response is highly reduced. But on this area, there's received the RPE scaffold transplant, you can see a large portion of the response is preserved. Here show a response from the baseline before, and here show after this RPE patch transplanted, compare with this empty scaffold without RPE, there is a large increase in amplitude. Just please note that there is difference in the scale. So although we see some kind of preservation, this response definitely is not, is much smaller than the normal eyes. So again, we this data has been published, and this is part of the package we submitted to FDA to start for a phase 1 trial to evaluate the safety in a human subject with transplant for these RPE cells. And finally, I like to acknowledge the Yichao and Aaron. They did the pig ERG recordings. And this is big project led by Capel Body and other research teams, to include induce the stem cell and RPEs for the treatment, and eventually now in a phase 1 trial. And I also thanks for this animal care people to maintain the animal. And I think it's one of the very critical part for good multifocal ERG recording is to keep eye stable and focused in the region. And they the veterinarians gave us a lot of suggestions on how to maintain the animal in a good condition with eye stability, which is the key. And also, I would like to thank you for your attention, and welcome if you have any questions.
Okay, yes, thank you very much for this interesting talk. Some questions? Or you can write later in the chat. If we have now not a direct question, we can go to the second lecture from Xiufeng Song. Please.
So I should have stopped sharing that. Yeah.
Professor John.
Okay.
Yes.
So let's start.
Please.
Wait a minute. So I have to first share my screen, right? Okay. Can you see my screen now?
Yes.
Yes.
Good.
Okay. So, wait a minute. Oops. Sorry, wait a minute. Okay. So let's start now. First, thank you very much for Dr. Joe and Ken for introduction and for warm invitation. I am very happy to have a chance to share our work experience with the Roland RETImap in our stem cell related research. So due to the time limitation, today my topic will focus on the performance of Roland RETImap in functional evaluation of chemical-induced AMD monkey model for stem cell therapy. So let's start with a short background. As we know, the AMD is the main cause of blindness in people older than 50 years old. This disease mainly involved in the macular region and lead to the central vision loss or distortion. With the increase in aging population worldwide, the disease has become a huge burden to family and society. This disease, in clinical side, can divide it into two subtypes, dry AMD and white AMD. For AMD at the late stage, usually this disease will lead the red cell loss, such as RPE cell or photoreceptor cells. Once these cells become lost or dysfunction, there is no cure treatment so far. So with the achievement of human pluripotent stem cell technology, now HPSC can differentiate into almost all body cells, especially including retinal cell. And as we can see, over the past decades, the retinal differentiation technology with HPSC actually achieved many milestones. First, we acquired RPE, RPC cells. Then later, we can also acquire retinal tissue like retinal organelles with the structure and morphology and molecular all very mimic the in vivo retina. This is our work. Firstly, achieved the functional retina with iPSC in vitro, and we also developed a protocol to acquire RPE cells with iPS or ESC. So all these cells have the similar characteristics as the cells from the in vivo. So these cells can solve the bigger issue, such as the seed cell source limitation. Therefore, the stem cell therapy to replace the lost cells has become an emerging treatment for AMD, that disease. However, before the stem cell therapy can be used to treat patients, the safety and efficacy have to be tested in animal models first. Therefore, we need to develop to disease animal models for stem cell therapy test. Recently, we have used chemical-induced retinal degeneration in rabbits and monkeys. This work mainly performed, contributed by my PhD student, Guan Jie. The work has been published. So we use the SMP and the induced retinal degeneration in rabbits. So we can see after the treatment, we can see the retinal tissue gradually degenerated. The thickness of retina and gradually decreased, and also the function we can see. So the another work we use the chemical-induced degeneration in monkey model also very nice. So when we injected SMP in the monkey the macular area, we can achieve a very lesion controllable retinal degeneration. So very mimic the AMD, that lesion. Over time, we can see after SMP treatment, the retinal tissue decreased. So how's the function? Why is it also cause the function dysfunction after the treatment? We very glad find the Roland RETImap, especially the multifocal. So we can perform the function test in the monkey and the rabbit. This is the in the monkey. So we in the rabbit, we performed the full ERG and the multifocal ERG. We can see after the over time, after the SMP treatment, the response is gradually decreased. And also this is the multifocal. So as Dr. Chen has showed a lot of details, here we will not repeat. So I'm very happy with the results, because the mfERG with the Roland map, we can really find what is true, since we can real-time monitoring the fundus and the stimulator. So this is a very nice data. And also we use this multifocal ERG to evaluate the functional change after SMP treatment in monkey. And so this is the instrument. So very nice, as you can see. So each area you can find that this is the normal, so control the monkey, and this is the SMP treatment the monkey. So this area you can see each angle, you can see the control is controlled very well and is confident with your data. And also the data can present it in a different way. This is the 3D image, and this is you can also get the circular area, since when the treatment maybe caused not generated the even damage. So you can directly check which area the wave is different. So you can do the analysis by the circular or the quadrant to get the statistical data. And this is the also we have used this. This is a multifocal ERG, so it shows clearly SMP caused the functional loss. This is 7 days after SMP treatment. So you can see the wave all become flat. Also, we use the this multifocal ERG that follow the long-term follow-up for the function changes, and we are happy. Okay, sorry. So this is the statistic. We do the statistic. This is 7 days after treatment, and this is pre-treatment. So can easily find after treatment the function amplitude and is largely decreased. And also we follow up the function changes. So this is the longest time we follow the 7 months after SMP treatment. We can still see the area with SMP treatment, the function did not reverse. Therefore, we have an animal model. We have stem cells. Therefore, we think that the stem cell move to the clinical treatment will not far, and will have a much promise for the blindness to reverse vision. Here a short summary. So we are very happy with the Roland RETImap. So this is a really excellent experiment and has a friendly internship, very easy to know and to handle, especially for the animals, and has a different animal analysis modes such as annual and quantums. Yeah, so this especially the mfERG can be monitored by infrared fundals. So ensuring the accuracy recording at the region of interest. I think this will be a very useful equipment for functional evaluation in animal models for gene and cell therapy. And so, finally, I'd like to thanks the members in my lab and to perform the experiment. And also thank you all for attention and welcome any questions.
Okay, thank you very much.
Yeah,
Very nice, interesting talk.
Thank you.
And one question I see is the ERG is under anesthesia?
ERG?
Yes.
Okay, could you please?
Is this performed under anesthesia when you make the animal multifocal ERG?
The ERG was performed, you mean so we performed the multifocal ERG for the monkey, and we performed the 4 ERG for the rabbit and multifocal ERG for rabbit too.
Yes, and you need anesthesia for
yes, of course, yeah, and that is right. Yeah, yes. This is important. Yeah.
Yes.
Yeah. Yes. And have to do the very nice the anesthesis, right? And also, as our experience, when perform the ERG, the environment it's very important. You have to control. You have to control this. Since we at the beginning of the experiment, we have we have met a lot of problem, but we are very thankful for the Dr. Tang help us resolve the problem, such as the environment noise. Yeah, thank you.
Yeah, thank you. Yeah, the signals are very, very small. And you had, it's important that all the noise from the room keep to very small as possible.
Yeah.
Yes. Okay.
Okay.
Now we come to the next lecture.
Yes.
Seeing you, and this was, now it's more pattern. Yeah, before was more flash ERG, multifocal ERG, and now we listen to the pattern.
Yes, but can you hear me?
Yes.
So probably Xiufeng has to stop the screen sharing first.
I think I have stopped, right?
No.
No, I did not stop.
So you only
Okay,
pause.
yes.
You only pause,
Okay.
not stop.
Okay, so did you say mine?
Okay.
Can you say mine?
Yes.
Yes.
Okay, good. Okay. Hi, everyone. So I'm Ying Xu from Jilan University. Today, I'm very glad to share my experience of working with RETImap to record the retina and the visual cortex light responses when we do the optic nerve transection on the monkey. So actually, compared with two previous speakers, they have a nice publications on this. I'm kind of preliminary. I'm kind of starting new on this project since I only started work on this project since last year. So today I'm going to show you some preliminary data on our recording. But I believe since I have working closely with my friends to set up this platform, so I believe I have some hands-on experience to share with you. You have some questions. So my project is to work on the optic nerve injury. So we know that optic nerve is important. That's actually composed of the axons of retinal ganglion cells that transmit signals from retina to the visual cortex. But the optic nerve can easily damage it or injured during an accident or traffic accident, or some like the children when they fight each other, they may hit the scar and may hit the optic nerve. So I took the project by setting up the optic nerve injury model on the monkeys so that we can apply like future application of the therapy, like a stem cell. So this is basically the protocols what we do on setting up the model. So on 1 or 2 weeks before the surgery, the optic nerve transection, we'll screen for the healthy monkey, and then we record the ERG response, and we will evoke the potential and do also the OCT and founder's image to find, and as well as the visual behavior.
I'm sorry, that your slide is not forwarding. So we are still on the first slide.
Really?
Yeah, now, but you are not in a presenting mode. It's just the overview mode.
What? Really? So
you need the slideshow mode. I'm not sure, but it's okay if we see. We can see now, but you also saw left panels.
But can you see this now? Can you?
Yes, we can see. Yeah, we can see everything. Yeah. Okay, that's fine.
Oh, okay. So shall I continue from here? So I just talk about setting up record the ERG and VEP, and also the visual behavior, and also do the OCT and the founder's image 1 week before the surgery. And then at different time points after the surgery, we did the same examinations. And sometimes I also add the functional, the MRI inspection on the monkey. So here is a illustration of the simple cartoon of the surgery we performed on optic nerve. So basically, we isolate the medial rectors of the monkey from nasal side, and we rotate the eye, and then we go deep enough, we can see the optic nerve. Then we use the scissor, we cut the optic nerve in half. We cut half of the optic nerve, and also you can see the contents of the optic nerve actually swelled, came out of the broken cells. So that means that indicates a successful build-up of the optic nerve transection. And then basically, today I'm going to talk more about the tech part. So then we use a 4-field stimuli from the Ganz field that enable us to record the light response from ganglion cells, which is represented by the PHNR, or photopic negative response under photopic range.
Now you're on.
Can you hear me?
Picture 4, slide 4. Yes.
This is, oh, maybe you want me to show like this?
So you talk now, slide 4, and then.
Yeah, I think you need to be in a slideshow mode right now. You just like in a file open mode. It's just not in slideshow mode. This is on slide 3. But you it's not in a slideshow mode. But we can see everything. But it's up to you. Then you need to move slide one by slide one.
Now it's, how about now? It's not slideshow.
No.
Oh, interesting. Okay, let me check.
Other screen.
你在下边，你下边有一个 slideshow 的那个 icon， 你点一下，要下边那一个。哎呀，就在那个。
点的呢，这真是全屏的，难道不是全屏的吗？啊，等一下，sorry。
你也许可能是按 show 的不一个Yeah.
How about this now?
还是没有。那你有可能是你那个 share 的那个另外你2个 screen 是不是啊？你可能 share 的另外一个 screen， 你可能必须得重新 share 一下。
Let me try to stop and restart again.
Yeah.
And I will share the screen try again. No. How about it now?
No, still the same.
No.
Yeah, it's good.
Now it's good.
Okay. How about now?
Yeah,
Now it's
good.
It's good?
Yeah.
Okay. So I continue with this. So we apply the 4-field stimuli and record the photopic negative response, which will represent light response from ganglion cells. And we can also use the 4-field flash to record the visual evoked potential that give us several waves, and that gives us the light response from visual cortex. But meanwhile, actually, since thanks to this SLO system, we can apply the pattern stimuli. Apply the pattern stimuli that we can record the pattern ERG and the pattern VEP from the monkey, so which enable us to also get a light response from ganglion cells as well as the visual cortex. So this is the operation, how we operate the SLO system. So basically, we will anesthetize the monkey. We use a crab eating macaque for this study. We anesthetize them and put them in front of the lens, and we cover one eye for the VEP recording. This is how we mount the electrodes. And then we adjust the focus knob, and so we look into the eye till we find a clear image of the macular region. So that's where we want to present the pattern stimuli like this. So after we apply the pattern stimuli, and then we can record the response from this region for the right eye and then for the left eye separately. So then we can analyze the data in this way. So this just show a raw data we record for a pattern visual evoked potential that you will give around like 10 to 20 microvolts response. And also we can get a pattern ERG recording, which also give like around 10 microvolts, which is relatively small compared to the A and B wave for the 4-field ERG. but it's clear. We can clearly see the waves. And so now your main result. So I will show you how we found that there is a decay of retinal light response after optic nerve transection, and also the visual evoked response, and also the degeneration of retina and the behavior decay after optic nerve transection. So first, we show so here shows the trace, the recording trace of the ERG, full field ERG, before the surgery, and at different time, at 2 weeks, 4 weeks, 8 weeks, on 12 weeks after the transection. And so this eye served as a sham control, and for the other eye, we have a real surgery. So from here, compare with these two eyes, you can see that both eyes will have a clear A wave and a B wave, and also under normal condition and before the surgery, have a clear PHNR wave. So we measured the PHNR peak, and you can notice from here, you can notice that for the surgery, so injured eye, the optic nerve amplitude becomes smaller compared with the control eye. So this is a statistic. This is the bar column showing the decrease of the PHNR amplitude in the injured eye. And then we also record the pattern ERG, and we measure the amplitude of both waves from N35 to P50 or P50 to the N95 wave. And before the surgery, both eyes have a liberalized response, especially for the second wave. But then after surgery, you can clearly see that both waves, especially the P50 to N95, decreased in the injured eye. And so here is a summary of these changes. But you may also notice, it's interesting to see that somehow the wave response started to recover after 2 months after surgery. So this is a pattern ERG. And then we go to the visual cortex. We record the full field visual evoked potential that enable us to see different waves like N1 to P1, and then there's a clear N2 to P2 wave under normal conditions or before the surgery. And then for the control eye, all the time we can see the clear 4 waves, 4 peaks in the control eye. But for the injured eye, you can notice that the second wave, the N2 to P2 wave, almost disappeared in the transection eye, especially after 12 weeks. So it's totally gone. Only 1 wave remained. So this is a summary of the changes. And then, if we apply the pattern VEP, the effect is even more dramatic. That in the normal eye, in the control eye, the 2 peaks, there are clear 2 peaks, which is remained all the time after the surgery. But for the injured eye, the P200 to N91, N135 peak, this almost disappeared. Quickly decreased after the injury. So here is a summary of the caves. And in this case, the VEP decreased all the time, even after 3 months after the injury. So basically, that's the data we recorded so far for the light response. And so, of course, we might check the structure of retina by the founder's image and also the MRI. For the MRI, the founder's image actually looks quite normal especially after like 2 months after the surgery. But for the MRI, we can see, it's hard to see here, there is some destructure of the optic nerve. And when we perform the OCT, we can measure the layers of RNFL, where the axons of ganglion cells locate, and also we measure the soma, the thickness of GCC, where the somas of ganglion cell locate. And here you can see that as time passed after surgery, the injured side, the thickness of both RNFL or ganglion cell somas decrease a lot compared with the sham surgery eye. So that means there is degeneration of retinal structure after the optic nerve transection. And we also do some behavior test to check the changes. So to do this, we use the optomotor system. So we set a screen and present the moving stripe in front of the monkey and track its eye movement. So the monkey can see the movement of the greetings, the eye will follow it. So then we change the width of the stripe, and to check how fast, how fine the stripes the monkey can follow the moving. And so here is the blue one is a injured eye, and for all the time, so the injured eye have to use a wider stripe to initiate its optomotor reaction. So that means the visual acuity is smaller, is impaired compared with the control eye. So that's the behavior. We also tried some other behavior data, which I'm not going to show here. But in summary, so our preliminary data shows that optic nerve transaction decreased light responses from ganglion cells, and also the VEP, that is the transmission to visual cortex. And the surgery impaired the retinal structure as well as the visual behavior. And since we are talk about this L system, and so by carefully compare with the Python ERG and with the full field stimuli, we think at least the change of Python ERG and the VEP results were very consistent with the full field stimuli result. And the second is we think the Python stimuli allow us to be better see the changes, so that is be more sensitive and also kind of stable. And most importantly is that since the stimuli, when we check the stimulation, check present Python stimuli on the retina, we focus on macular region, so that enable us to use this system to record the response from region of interest, just like as Xiufeng said that. So basically that's all for my presentation. So I would acknowledge my team and also Professor Hui Yang and Xuan Wei Liang from Zhongshan Ophthalmological Center for this. And so basically that's the end of my talk. So do you have any questions?
Thank you very much for this presentation. Some questions?
It's very interesting. Yeah, just a quick one. I missed where this VEP is recorded. So you have one eye is transected. Do you record VEP from two separate left hemisphere or right hemisphere? And then if you have seen any difference from two hemisphere, because the eye is projected on both sides.
So yes, so we covered one eye, and record the VEP response from the other side.
But from the cortex, what region is it combined of both cortex, or just you can record from left cortex versus right and right side cortex?
For the recording, no, actually we record both sides, but since we only injured one eye, so can we compare the data?
Yeah, but one eye projected to both left and right hemisphere, isn't it?
Right, yes, but the collateral side contribute more than the ipsilateral side.
Okay, thanks.
Some more? Some comments? Mr. Flangtan?
Yeah, the doctors ask if the monkey can lead to the refraction, the optical refraction before the PERG and the PVEP. Yeah, I think it didn't. Yeah, because we use our SLO to adjust the refraction.
Yes, and if when the SLO image
Yeah.
is correct, then the
Yeah.
stimulus presentation on the retina is also sharp.
Yeah, and this the one Dr. asked the mfERG is performed to awaken animal or the under anesthesia. I think he's under anesthesia.
You have to exercise the animal. Yeah.
But VEP is difficult with anesthesia. You need special, yeah?
Not actually for VEP, we don't want to dilate the pupa. So you want, I mean, you want to anesthetize too much, or else the pupa will be dilated.
Okay. Then thank you very much for all people to join our webinar. Thank you very much for the very interesting lectures. And I think we will continue. And when we prepare the next lecture, you all time will get with the newsletter, the information. And for the time zone, I think now we have all over the world in one time zone. In US is very early, and in Asia, China is all very, very late. But we all together in one session is possible. Thank you. And see you,
Thank you.
on the next lectures.
Okay.
Thank you very much.
Okay. Bye-bye.
Thank you. Bye-bye.
Bye bye.